This is the Questa project 2014 experiment A done by group 2A of One Integrity. The group members are Chik, Yolene, Candice, Irfan, Sofan, B, Zufri, Brendan Parkinger, and Tan Wei Lun, Nikki. The purpose of this experiment is to investigate the effects of different light intensities on the rate of photosynthesis.
Introduction. This experiment is about investigating the different light intensities on the rate of photosynthesis on hydrilla plants. Photosynthesis. Photosynthesis is the sequence of reaction performed by green plants, algae, and photosynthetic bacteria in which light energy is converted into chemical energy and used to produce carbohydrates and ultimately used to produce all the materials of the plant. It can be summed up as 6 molecules of carbon dioxide plus 6 molecules of water plus light energy equals to a molecule of carbohydrate plus 6 molecules of oxygen.
The materials used for this experiment are a 500 cm³ beaker, a retard stand and a clamp, a boiling tube, a table lamp, a meter ruler, a dilute sodium hydrogen carbonate solution, a freshwater plant, in this case is a hydrilla, and a tripod stand. Procedures. A piece of 5 cm long hydrilla plant was cut. The 500 cm³ beaker was filled fully with water and placed on the tripod stand. The boiling tube was filled with dilute sodium hydrogen carbonate and the hydrilla plant was placed in it. The boiling tube was clamped and placed in the beaker. The thermometer was also placed in the beaker. The lamp was placed at different distances of 50 cm, 40 cm, 30 cm, 20 cm, and 10 cm away from the boiling tube. Firstly, the lamp was turned on for a time of 1 minute. Then, the lamp was turned off and the number of air bubbles, which is the oxygen produced by the plant during photosynthesis, was counted. This was then repeated twice for each distance. These are some of the pictures we took before and during the experiment.
These are the results we acquired. Discussion. We observed that the number of air bubbles produced by the plant were not dependent on the distance from the plant to the lamp, whilst in the theory, the amount of oxygen produced by the plant should be dependent on the light intensity. There are many factors that could have caused this, some of which are present in our experiment.
Some examples of them are the pH level of dilute sodium hydrogen carbonate solution may have affected the rate of photosynthesis. Plants are also less receptive to yellow and green light, and they are most receptive to blue and red light. In our experiment, the color of the light was yellow. This meant the rate of photosynthesis was lower. The water given may have also contained some impurities like chlorine and fluoride. The room was filled with light, and that the lamp may have no effect on the plant. In conclusion, we can tell from the relationship between the number of bubbles produced and the intensity of the light that photosynthesis was not affected by the intensity of the light, whilst in theory that it should have been affected. This could be due to the errors neglected by both parties during the experiment. One of the possible errors are the bubble produced by the plant may not have been filled completely with oxygen, but instead filled with some carbon dioxide from the respiration of the plant.
Other factors neglected would include the factor listed in the slides above. The experiment also did not include a control setup, which is vital for any experiment to produce a conclusive result. Thus, even if we were to have a result, it would have been an inconclusive one as proven by the factor we listed, and we would not be able to put it into use either ways. These are the references. Thank you and have a nice day.